All right, good morning, everybody. Give people another minute or so to uh, wander in, get their morning coffees, that kind of stuff. Get out of bed, or maybe even not get out of bed. It doesn't really matter, to be honest. All right, so before we start going through the flow of genetic information PowerPoint, does anybody have any questions, concerns, things that they want to ask me before we get started? I'll try and do this every class so that you have a chance to kind of go back over material that we studied prior to this class. Don't really have an awful lot to go over yet, but we will do in the goodness of time. Let's get rid of that. Let's make sure I know what we're doing of. All right, well, will anything today be on exam one? Maybe. So let me get up crab sticks. Where's that gone? No. You don't want to look at that. Sorry. That's what I meant to press. I got so much stuff open on my computer. Let's see. So, tricky question to answer, obviously, because I'm not going to tell you exactly what's on the exam because that would defeat the purpose of it. But essentially, what we're going to be doing today is doing a kind of recap stroke overview. So a lot of what we're going to be talking today will be covered in far greater detail in their respective sections. So there is basic information that you need to know on here. But to be honest, a lot of the information's kind of basic enough. Yeah, I don't know. I've got to be careful about what I say, I guess. To be honest, one of the main things which you'll find on here, which we're not going to cover in great detail later on, are kind of the different types of mutations, things like that. So that's something that's worth being familiar with. But again, I'm not a person that will grill you incessantly on, minutia, tiny facts, things like that, because it's kind of pointless because you have all the information here. You'll be able to answer those questions real easy. So really, I shouldn't worry too much. We'll get really into the meat of things when we start on Mendelian genetics on Friday. This is really just going back over stuff and getting you in the right frame of mind, essentially. Yeah, for sure, Zuleika. Just interrupt whenever is a good moment, and I'll go over whatever it is you want to ask about.
Okay, sounds good.
All right, cool beans. So essentially, the purpose of the purpose of this PowerPoint is really to refresh your memories in a major sense, right, from GenBio 1, because it may have been some time since you took that. And these are pretty central concepts. Foundational stuff really is the best way of putting it for a lot of the material that we'll be studying later. So these are things not necessarily that you will be grilled on in the exam, but they're things which if you don't understand, then the stuff that you do learn about later will be very difficult to understand, right? So this is a good starting point really in the course. And so some of this may be stuff you already know, in which case that's awesome, right? Because that means you already know it. I don't have to worry about bringing you up to speed. But if you don't, then that's what we're doing here, essentially, right? So some of this may seem a little bit duplicative, right, over what you've learned before, but that's fine, right? That's kind of part of the idea. So essentially, whenever we talk about genetics, the very first thing that we start with is DNA, right? Because that's by large part, the heritable material. Oh, okay. I was getting weird hand gestures from my wife. Didn't know what she was telling me. And there's a few different things about DNA which you should know and which are really important about a lot of other stuff, right? One of which is that obviously, this is the structure of DNA, but the structure is what's the best way of putting it? So it's a repeating structure, right? And each nucleotide or deoxyribonucleotide, if we're being specific, consists of a phosphate in a sugar, right? Which gets joined together to make the phosphate sugar backbone, right? This is the kind of like the structure of DNA, right? It's not the information. The information is encoded by these nitrogenous bases, right? And these nitrogenous bases are what are responsible for the information, right, in DNA, right? This is the information, these 4 bases, adenine, cytosine, guanine, and thymine. And also for the hydrogen bonding between the opposing strands, right? So hopefully you'll know, how hydrogen bonds form. Actually, here's a question. How do hydrogen bonds form? What is necessary for a hydrogen bond to form? Questions to the class. You can type it in or you can shout it out. This is a bit of a, oh crap, what did I learn back in Gen Bio 1 all those many moons ago? What do you need for a hydrogen bond to form? I know you're there because I can see you in the class list. Okay, I'll give you a clue. It's there's 3 particular atoms. 4 maybe. Depending on how you look at it. I'll give you one. You need a hydrogen, right? What else do you need? I'll give you another freebie. Fluorine, right? And you're probably thinking, like, what the hell? Fluorine? We don't ever see that, which is good because it's a nasty ass atom, particularly when it's attached to a hydrogen. What else do we need? There's a bunch of them on there. Oxygen? Yay! Sorry, that's that. I'm not being rude. Oxygen, and what's the other one? There's a whole bunch of them in those bases. It's kind of in the name of them. Nitrogen, right? So nitrogen, oxygen, fluorine are extremely they're electronegative, right? They love electrons. Yeah, thank you, Lee. And when they're bound with a hydrogen, they form what's called a polar covalent bond. There you go. Oops, let's do this properly. Half my chat is off the screen, so I've got to make sure I'm actually typing correctly. There you go. So, all right, so let's kind of expand on that question. How many, if that's what you need, right? So you need a nitrogen and a hydrogen, or an oxygen and a hydrogen, or a nitrogen and something else, right? Let's say. How many hydrogen bonds is adenine able to form? Right, it's going to be from this side, right? So this face of the base, essentially. So you might know this just, oh, shit, I hope you're all right, whoever iPhone is. That's a pretty terrible thing to have happen. Back to the question. Oh, Lauren. Okay. Well, I hope that you're safe. You're not injured. If you are, there are other things that are more important than attending genetics lecture. So how many nitrogens and hydrogens or nitrogens or something else do you find here in this face of the base? It's going to be 2, right? Well, there's 1 nitrogen on its own, and then there's nitrogen and hydrogen here, right? So essentially you're going to have 2 polar covalent bonds in this region, give or take. Nitrogen is kind of funky as well. Does all kinds of weird things. To be honest, Lauren, I would go do that now. You can watch this later. I would go to the, well, if you can, go to the ER now and get checked. We were in a traffic accident a while back too. All 4 of us actually got rear-ended on Culebra Road. And we all ended up with concussion and whiplash, right? So it's good to get sorted out ahead of time. Also really important if you end up engaging a lawyer too, which we also did. So go do that now and, catch up on this stuff later. It's a lot more important. And besides, I don't want to lose any students, right? I mean, I've got a record to keep here. Not 1 student has ever been lost in my class, right? So it's personally important to me. Obviously I care about you as well, but yeah. So go and get sorted out, and also email me if you want, and I'll give you whatever advice I have about dealing with stuff like this. It's a pretty unpleasant situation. Anyway, so adenine can form 2 hydrogen bonds. Right, and now we look at guanine. How many hydrogen bonds do you think guanine is going to be able to form? Right, again, look, how many polar bonds do we have here? We've got oxygen and carbon, that's a polar bond. Nitrogen and hydrogen, that's a polar bond. And they've also got nitrogen and hydrogen here. That's also a polar bond. Pardon me. That gives us 3 hydrogen bonds, right? So we now know that adenine and guanine can't base pair, correct? Right? So we also know that cytosine also has 3 potential bonds, and these 2 are different in size. So just these properties, right? You can just look at these things and figure them out just on first principles, essentially. What are you doing, dog? My dog is at the alert. It's really funny. She sits like a person sometimes. Luna. No, she's keeping her eyes open for something. She's probably waiting for the old lady across the road to come out in her dressing gown and start barking at her. It's the only person she ever barks at is the cute old lady across the road in a dressing gown when she puts the wheelie bin out. Yes, she's very funny. She's super squishable as well, which is also nice. She's like a stress ball. Anyway, so just from first principles, you can get a decent understanding of how these bases fit together, right? And this is extraordinarily important because this is the foundation of a couple of really important things. One is the fidelity of DNA replication, right? How accurate DNA polymerase is in adding the right base opposite the correct base, right? Without which you would be basically every time your cells divide, it'd be a gibbering mess, right? And so you don't want that to happen. And also, this is how we transcribe DNA into mRNA, right? Through this base complementarity. Obviously, thymine is changed to uracil, right? That's one of the main differences between DNA and RNA. But the essential principles remain. So that's one really big deal, right? The other big deal is DNA has directionality, right? And the reason for this is, and so lipids don't have directionality, but lipids are real funky ass macromolecules, to be honest. Glucose, starch, cellulose, things like that do not have directionality, right? They're essentially the same one end to the other. but DNA, mRNA, and protein do, and that is really, really important. And so that directionality comes from the fact that DNA can only be added, or new nucleotides can only be added to this 3' hydroxyl group at the bottom, nowhere else. They can't be added to the other end, right, for a bunch of different reasons, which we'll get into later. And so this directionality has a lot of, again, it's really important in DNA replication because you can only add new nucleotides at the 3' end. It's also really important in transcription and translation, right? Because your RNA molecule is only going to be synthesized also in one direction. And that kind of leads us to some fairly trippy kind of concepts to get your head around. And because mRNA is read, it's translated in one particular direction only, then you can form your polypeptide only in one direction too. Right, so even though I'm sure you know all of this already, it's kind of like information that it's like knowing letters, right? You know, without knowing letters, you don't know words. Without knowing words, you can't speak or read or understand what people are saying. It's like a very fundamental piece of information or knowledge that you need to know about genetics in general. Pardon me. I think I had my dog sleep with us last night, so my ability to breathe is severely curtailed. Nothing much new here that you don't already know. I've already talked about base pairing. You already know, hopefully, that DNA is anti-parallel. Let's move this over here a little bit. Which, come on, get you back there. I don't need to know what I look like. I'm just kind of moving my image of me across the screen. The main purpose for having that there is to make sure I don't get cut off at the nostrils or something. So anyway, the anti-parallel deal basically refers to the fact that one strand going 5 to 3' up here, the other strand is going in the opposite direction, right? So it's going from 5 to 3' down the page instead. And really, if one were to be really anal retentive about it, and I have been in the past, for full disclosure, the letters of the strand going up the page should be upside down, right? Because it is truly anti-parallel. It's not this the bases get twisted around; they are upside down relative to the other. So if you have a T like so going up, on the other side you have an A going down, for example. But one of the things I want to show you from this image, which you may or may not know, is that DNA also has topography. It has shape to it. It's kind of like the difference between the hill country and, I don't know, Corpus Christi, right? We have topography, Corpus Christi does not. At least every time I've been through it, it hasn't. Maybe if you go on the overpasses, it does. And so DNA has what's called a major groove, which is this larger spacing between the helix. It's not like two bits of wire twisted together. It's kind of, essentially, it's like two bits of wire which are kind of looped around something else, right? Like a pencil, I guess, would be the best way of thinking of it. And it has a minor groove where those two strands are closer together. And this is really important for a bunch of proteins which bind to DNA, right? So not only do they recognize the sequence of DNA, right, these individual bases, but they also recognize the shape. I think from memory, I think it's TRPR, the tryptophan repressor, which we'll learn about in a wee bit. Actually, it's probably near the end of the course, to be honest. TRPR binds to the tryptophan operator and the tryptophan operon, and it recognizes from memory the two major grooves in that sequence of DNA, right? So it doesn't just sit flat up to the DNA. Essentially, the DNA is grooves, it comes in and it locks in to the major grooves, right? It doesn't touch the minor groove at all. Kind of hard to do with the hands, but you get the idea, right? So keep that in mind as well. So essentially, one thing that I want to really get across, and this is super important too, is that every cell has the same DNA. Let's go to screen share, which is a little bit more useful. And right, so these two different cell types, they are, this one's a neuron. These are keratinocytes, which are, I think, skin cells or skin or hair cells. Basically, they're structural cells. You could even say they're squamous, right? Which means they're flat and pardon me, the edges stick together. All right, chat's gone and disappeared. If you got any chat questions, just go cough and I'll go back and get that. And these are really pretty pictures. I'm a microscopist at heart, I guess, even though I'm a geneticist as well. And I love microscopes. They're just absolute fabulous things to use to explore the world. I like the really small world, anyway. And these images, I think this one, but certainly this one, are taken with what's called a confocal laser scanning microscope. So rather than having a light shine up through the sample into your eyeballs, this actually uses a laser, and it scans basically it's a 3D object or subject, right? And so it scans back and forth in a XY fashion, right, until it's scanned over the entire field of view that you want to look at. And then the sample gets raised by a fixed amount, usually, I don't know, 10 microns, something like that. And then the whole process is repeated again. So every single part of that subject is in focus, which is not the case when you use like epifluorescence microscopes, when you have the light shining up through the bottom. You get a lot of scattering of light. This is a big problem that I have with my worms because they are three-dimensional objects. And so that allows you to get these most fabulously detailed, truly beautiful pictures of three-dimensional images or objects like this cell, right? And so this is this must be a pretty, this is probably a neuron that's grown in vitro, not in vivo, because in vivo they'd have a very different picture. But essentially, the green is a protein called actin, which is a cytoskeletal filament, typically used in here in growth cones or kind of to give structure to the cell. And the red is the red. Oh, microtubules, right? So those are also a cytoskeletal filament, and those are used to transport stuff. So if you always wonder how does the stuff get from basically the bottom of your spine to your, I'm not going to lift up my foot and show you my toes, to your toes, it's going to be down these microtubules. Anyway, so I'm sorry, I'm kind of like nerding out a little bit about these pictures because they're super pretty. And so the actin is labeled with GFP, which is a green fluorescent protein. And my guess is that the microtubules are with DS red or M cherry or some other red fluorescent protein, fluorescent protein or label, whatever it might be. But anyway, getting back to the point at hand, this cell here, neuron, has a very specific function, right? It's to carry electrical impulses from one part of your body to another. And these cells, which are structural cells, they're either in your skin or in your hair, or maybe even both, I'm not 100% sure, have exactly the same information in them. exactly the same, right? There is not really any significant difference between these two cells in terms of which genes they can express, right? Because they all have the same information. It's a really important point to get. The big difference, what makes them different, is what genes they do express, right? And so these genes here, these cells here, obviously are going to be expressing different proteins in different places. And that is what gives them their identity, compared with these cells, which express a different array of proteins. Obviously, some of those overlap because all cells need ribosomes, for example, to be able to do translation. But others will be very specific to those cells. For example, these cells will be producing probably neurotransmitters, right, to be able to propagate that signal onto the next cell or neuron. These cells probably produce a lot of collagen, right, which is a structural, actually, I'm not even entirely sure what collagen is. Glycoprotein, I think. Anyway, which is necessary for it to be able to maintain its structure, right? So gene expression, or really differential gene regulation, which we'll get to in considerably more detail by the time we get to PowerPoint 18, is what makes things different, right? And this is really, let's go back here, a case of controlling where genes are expressed, right? You have different genes expressed in your liver compared to your lungs, right? When those genes are expressed, you know, some genes will need to be expressed in your early development. I'm trying to get the chat box back. God damn it. Zoom is a real pain in the posterior. No, it's not letting me have it. Let's see if I can go back. Well, there it is. Oh, yes. Thank you, Zuleika. I do know that's because it's a subject that oh crap, chat has gone again. There's no chapter 14, not because it's like one of those flaws that doesn't exist. It's because the way I teach the course, I'm just not using that PowerPoint for a bunch of different reasons. So don't worry about it not being there. It's not an error in the numbering. It's just a PowerPoint that someone else uses that I don't. Sorry, I have allergy issues. Right, so some genes are expressed in different stages of development. Some are not expressed in development at all and only expressed in adults. And different amounts and in different contexts too, right? So, you know, if you are old enough to have a drink, then when you do, your liver will go, oh crap, out ethanol, you know, it's like a toxin, it's really bad for you. And it will produce, you know, enzymes which will break ethanol down into something less harmful. So all of that is done by essentially the control of gene expression. And when we talk about gene expression, what we really mean is this process. This is called the central, well, it's actually not called the central dogma of molecular biology. That's a bit of a perpetuated mistake. This is the sequence hypothesis. Central dogma is it doesn't go backwards. Some of you have had me in Gen Bio 1 know that's like a constant like rant of mine, I guess. But anyway, and so really gene expression is this whole process, right? Essentially going from DNA, which is it's really an inactive storage molecule, right? It's like having books in the library. It doesn't do anything itself. And converting that information into stuff that does stuff, right? And typically that's protein. It can also be RNAs, right? So ribosomal RNA is an enzyme as well, called a ribozyme. Give me just a second. Sorry about that. Yeah, so RNA is a ribozyme. It's an enzyme that catalyzes peptide bond formation. And these intermediate steps, which are somewhat simplified here because there's an awful lot more going on than just this, as you'll see later, are the processes by which that happens, which is typically transcription and which is the production of mRNA from DNA. Or rather RNA, sorry, I have to be careful here because you can have what are called non-coding RNAs as well, such as rRNA, tRNA, and many, many others. Messenger RNA, however, is the one that carries that information to the protein synthesis machinery, the ribosomes. In prokaryotes, that's just floating around in the cytoplasm. In eukaryotes, that's actually outside of the nucleus, so it serves a messenger function too. And then obviously we turn that information in the mRNA into a polypeptide sequence in the protein. So when we talk about gene expression or central dogma, I typically don't refer to it as a central dogma for various reasons, but when we talk about gene expression, this is what we're talking about here. Just double checking the for some reason, when you share your screen, let's see if I can go back to sharing just the PowerPoint. When you share the screen, you can't see the chat box. Let's see if I can get it back. Yeah, got it back now. Yay. Slowly figuring Zoom out. It's better than WebEx, which is a giant pile of steaming turds as far as I'm concerned. But it's still got its issues. So transcription is essentially consists of various stages. We're going to talk about this in more depth later. It's really the starting the process of transcription, right, which is down to these regulatory sequences, and the actual mechanics of producing the mRNA transcript or the RNA transcript, right? Sometimes if I say mRNA, I basically mean all RNAs, but because we're talking about gene expression, typically I'm talking about mRNA, just to clarify that. And this is a really cool picture because there's a lot going on here, right, which may not be evident at first pass. So obviously, this is our DNA double helix. It has to be separated, right, to be able to use that information, right, to be able to access it. The second thing is that you can see that these two strands, this DNA RNA hybrid, they're base paired. Right? Remember what I was talking about earlier about that base pair complementarity. And if you look real carefully, you can actually see that this strand here is going in the opposite direction, right? So the whole rule of the anti-parallel double helix still applies. So this mRNA strand is being synthesized in a 5 to 3' direction, right? So it's going up the page. You can only add new RNA nucleotides at the 3' end, right? So eventually this whole fork will progress up the page and that mRNA strand will get longer and longer, right? It won't stay attached for the whole stretch, but where it's being synthesized, obviously does, right? You'll also see that there are no T's, so these are all uracils, right? So all of those, instead of adding a thiamine opposite the adenine, a uracil gets added instead, right? So that's another difference. RNA is actually a pretty weird and wonderful molecule, to be honest. Now, the real tricky thing to get is that the information being copied is actually in this strand over here. So when you have transcription, you have essentially two strands of DNA, both matter. One is called the coding strand. That's what contains the information. The other one is a template strand. That's the one that's used to make the mRNA. It's a real kind of a pie-eyed kind of concept to get. But if you look, compare these two, right? So this is actually kind of a neat way of doing it. It's a little bit kind of in detail, but hopefully it will help get the point across. So essentially, look at here, right? So we've got a T in this strand, uracil in this strand, A, space, because there's an overlap. Thymine, uracil, C, T, U, C, so on, right? So this mRNA strand being synthesized on this side, on the left-hand side, is, other than there being uracils in place of thymines, is an exact copy of the coding strand on this side, right? So the actual direction of the gene is up the page, right? So this gene goes from down here up the page, up this strand. This is the coding strand. We'll come to that later. So this, essentially, I'm kind of putting a worm in your ear so that when we come across this concept next, then it won't be as much of a surprise, essentially.
I
Anybody?
had some. Oh, I'm sorry.
No, go for it. Oh, questions about transcription.
Yes. So I was wondering, with transcription and the audio recording that you like provided for us, you
Hey,
said
you listened to it.
Yeah. You said nucleic acid was with nucleic acid, and for translation, it was nucleic acid and protein. So I was just wondering if with transcription, there's like no mismatching, and with translation, it was only mix matching. Like it can't be the same, right?
Oh, well, so I recorded those a little while ago, so hopefully I can
Okay.
remember what I was talking about. But essentially, DNA and mRNA are both nucleic acids, right? They differ in two main ways. One is that DNA is deoxy. What that deoxy means is that an oxygen is removed from, hopefully you can see what I'm pointing at, this carbon here. So the second prime carbon in RNA has a hydroxyl group too. So RNA has two hydroxyl groups. DNA only has one, right? And also thymines are changed to uracil. But other than that, they're exactly the same, right? So in terms of transcription, what you're doing is you're copying the DNA into a different but related form, which is mRNA, right? So you're not really changing the overall format of the information. The information is more or less largely the same. Pardon me. Whereas when you go to translation, here we go. Now you're taking that information in nucleic acid form, and you're turning it into not more information, which is DNA to mRNA, but you're turning it into something functional, right? By putting together these series of amino acids into this polypeptide chain and then a functional three-dimensional protein. So that's, in one sense, one way to remember what transcription is versus translation, right? Transcription is that you're copying. It's like if I gave you some text to write out, right? You know, you're copying. You might put like little heart shapes over the eyes, or you might just put dots or whatever. But by and large, the information is exactly the same. Just the same as when you hand out a copy of your transcript, right? Your transcript is a record of all of the courses you've taken and your grades, right? It's a copy. Now, if you wanted to go study in Russia, right, you'd need to translate that into something that a Russian could understand, you know, presume,
Oh, okay.
right? So translation is really it's literally translating one form of information into another form, right? Into another thing, right? And the the thing that does the translation are these what are called tRNAs here, right? They're the ones that it's like the Rosetta Stone, which allowed people to translate hieroglyphics into uh, modern day languages, right? These are the intermediary which turn this information into this amino acid, essentially, right? So that's
Okay.
really what I mean by that.
Okay, that makes more sense.
Yeah, cool, good question.
Thank you.
So, we don't need to know that. So one of the things, and this is if you remember back to when is it, Monday, when I talked about before the structure of DNA was elucidated, people used to think that proteins must be the hereditary material because they're more complex than DNA is, right? And so DNA or mRNA consists of 4 bases, nothing more. Yet we have 20 different amino acids, right? And so how do you go from 4 bases or 4 distinct bits of information to 20 amino acids? And really the analogy would be the same way we go from 26 letters in our alphabet to hundreds of thousands of words, right? We group together those letters into words. Now, in biological terms, it's a bit simpler than language, and those words are 3 letters long. Right, so this is what's called the genetic code, which is really, if you were, a ribosome, you'd be like, okay, that goes to that, that goes to that, that goes to that, right? This is how you would translate mRNAs into amino acid sequences, because every single triplet or codon, right, which is the formal name, codes for a specific amino acid, right? And so if you look down here, essentially we have 4 bases to the power of 3. That gives us 64 codons, which is more than enough to encode the information for 20 amino acids. And that excess essentially leads to, in many cases, multiple codons encoding for the same amino acid. And that is a phenomenon called degeneracy. Right? It doesn't mean that these codons are slouching around up to no good. What it means is that quite often the last letter of that codon does not matter, right? It's the first 2 letters which specify the amino acid. Not always, as you can see between phenylalanine and leucine here, but certainly say proline, as long as you have CC, you got proline, right? And that's essentially a buffer against mutation. Okay, I'm not going to get into that because I showed you that already. Little bit of a kind of intro into RNAs. RNAs are funky, right? So if we're kind of anthropomorphizing, DNA would be the staid, resolute individual that never changes, isn't very interesting. RNA would be like super funky, up to all kinds of different stuff, constantly changing their fashion, let's say. And one of the things is that RNA can also base pair, just the same as DNA does. DNA forming obviously the double helix, but the RNA can base pair with itself, right? And so what that means is that it can form lots of different structures, right? So this kind of squiggly format for the tRNA is not a slip of the wrist of the person doing the illustration. It actually can form as a result of these stem and loop structures, right? So you can see areas of, I don't even know what this one is, to be honest. But this RNA structure, we have some double-stranded RNA here. We have some loops over here. We have all kinds of funky structures. Okay, so just in the last 5 minutes of class, just so I make sure we get through this, we're going to start on Mendel on Friday. So I want to kind of clear the shop, so to speak, before we start on that. Sorry, dog being distracting. So essentially, mutation is the only form of genetic novelty, right? This is something that cannot be overstated, right? All of us are what we are due to random mutations occurring over our evolutionary history, right? So in the case of Homo sapiens, that's what, I don't know, like 60 to 80,000 years. In the case of Homo species, I get a little bit fuzzy when it comes to dates. I think that's about 250,000 years, give or take. I can't remember exactly when, can't remember what the first Homo species was. It's not Homo erectus. I don't know, can't remember. Anyway, there's a whole bunch of them. But essentially, we are the way we are because of all of those random mutations which occur either during DNA replication, through what I like to call external insults, right? So it's not people going rah, 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 rah to our cells, but things like gamma or UV radiation or chemical mutagenesis or whatever, right? That changes our DNA. And those changes must be inherited, right? So changes that occur in your DNA in your somatic cells, they die with you, right? So, I mean, I guess the harshest way of thinking about that is that, you know, if someone gets cancer, then they die of that cancer, right? But their children do not, right? So those changes have to be heritable, which means they have to be in your germline, you know, whatever cells you'll be using to produce the next generation. And those changes must change the fitness somehow of those offspring. Because if you think about it, this is something that, because I'm not really much of an evolutionary biologist by any stretch. I kind of dabbled in it for a while, but it's not really my cup of tea. But something that really struck me is that if you think about this, the first mutation is going to be one in the whole population. So if, well, I finished having kids now, but let's say I hadn't. If I did have a heritable change, I would be in the US 1 in 350 million, right? That would be the frequency of that mutation or that change, right? So over time, something has to act to sustain that, because otherwise it will just disappear, right? And so really when we talk about mutations, obviously they can cause diseases. Typically they don't make any changes at all, and we'll get to why in a bit. But really what we're talking about this is a hand-drawn picture, I know, but it's a really neat picture. When we talk about mutation, what we mean is that these are genetic differences in a population, right? We are all different subtly in small ways in terms of our differences. And to be honest, San Antonio is very different, right? Because it has people from all over the Americas, right? It has people from Central America, has people from Mexico, has people from the East, from Europe, essentially, has people from that originally came from Africa. You know, people come from the north of the US, right? There's a lot of genetic diversity in just this region that we're in. Now, obviously, we're not all foraging around for grubs and beetles and getting eaten by saber-toothed tigers. But essentially, that genetic diversity is going to be acted upon by natural selection, right? And natural selection can be thought of as a funnel, right? It's going to concentrate down some particular variations, and it's going to discard others, right? Those which reduce your fitness are going to get lost. So if you have a mutation which causes you to run slowly, you're going to get eaten by a saber-toothed tiger, right? And the ones that don't have it are not, right? As they say, it pays to be the second slowest person being chased by a tiger, right? And so eventually over time, and this is a big process of time, that allele frequency, right, that form of that gene which arose through mutation up here is going to increase in frequency, right? And that's going to result in improvement of a particular trait, improvement of the fitness of a population. And if sufficient numbers of those changes occur, then you may end up getting speciation, right? The difference between Australopithecus, let's say, and Homo whatever, right? Who shared a last common ancestor. And just more for your information than anything else, we have a whole bunch of different ways of classifying those mutations, right? So it depends very much on what effect those mutations have, right? Which can be effect on translation, could be effect on function, could be effect on expression, where those are, what the molecular basis is, things like that. So don't get too wrapped up in all these various details, but they're worth looking through just to give yourself that kind of, overview, because we will be referring to some of these things later on in the course. And I've gone over a couple of minutes, but that's pretty much all I've got for today. We'll be starting on Mendel on Friday. So look over that PowerPoint or even listen to it, which is really cool. I'm so proud. I've got someone who's listened to one of my audio files. And 2, go through any of this and make a list of questions of stuff that doesn't make sense, right? It's a good habit to get into. And that's pretty much it from me. So for those of you that I have for lab, we'll be back on Zoom in about a coffee's time's worth or 2 at 11 o'clock. And for everybody else, have a fabulous day, and I shall see you next time I see you. And good luck to Lauren as well. Hopefully you get sorted out and, everything works out okay. All right. Have a fun day. Hang on, quick question from Marco. Give me a second, Marco. Which one is this? So homework 1 is due on Saturday midnight, 29th. And, oh, I see what you mean. I'd have to go double check to see exactly what the homework is on. But it's all the kinds of language and stuff that we're learning at the moment. That's essentially homework 1. Some of the intro stuff just to get you up to speed. So hopefully that helped. Cool. All right. Catch you later.